Thank you all for joining our webinar, Non-Invasive Clinical Biomarker Discovery and Development with the Breath Biopsy Omni Assay. Our goal is to give you a better understanding of breath as a rich new source of biomarkers that can be reliably detected and measured using our breath biopsy system. Before we jump into that, some quick background on who we are. Alstone Medical's mission is to save 100,000 lives and $1.5 billion in healthcare costs. And the way we plan to do that is by using breath biopsy to develop diagnostic tests for early detection and precision medicine. We have our own programs currently focused in respiratory diseases, liver disease, lung cancer, and digestive health. And we also collaborate with pharma, biotech, industrial, and academic partners across a wide range of disease areas to address their specific research or clinical questions. Here are some examples of the disease areas we have worked in, along with our collaboration partners. And on the right are some illustrative results broken down by application, either early detection or precision medicine, along with the patient numbers and performance. So what exactly are we measuring in exhaled breath? We are measuring the volatile portion, and more specifically, volatile organic compounds, or VOCs. These can come from two sources: endogenous, where they are the end products of metabolic processes and are affected by both physiological and pathological mechanisms, or exogenous, where they are introduced through external factors such as diet, drugs, exercise, smoking, vaping, and the microbiome. A breath sample, therefore, represents a snapshot in time of a patient's phenotype and can be used to non-invasively monitor patient groups as they progress through various exposures, diseases, or treatments. Breath analysis doesn't just report on the lungs, it offers the opportunity to collect a sample that is representative of tissues throughout the whole body. VOCs can come from not only local cellular metabolism, lung tissue, and the lung microbiome, but also systemically, as VOCs exchange readily from circulation into the lung alveoli and can be measured on exhaled breath. This slide really highlights the utility of breath to provide useful biomarkers outside of just respiratory research. If we look at the cumulative number of clinical trials involving breath analysis, there are a broad range of therapeutic areas covered. Why breath? We think there are quite a few compelling reasons to consider breath analysis. It's completely non-invasive, pain-free, and well-tolerated. Unlike with tissues and biofluids, there is an inexhaustible supply. VOCs represent what is actually happening phenotypically, providing a dynamic view of current metabolism and disease biology. Our process that I will describe later allows us to identify the VOCs present rather than relying on pattern recognition, which can be difficult to relate back to underlying biology. By identifying the VOCs, we can relate these to biological processes. Our process allows for pre-concentration, so detection limits are in the parts per trillion range. This is particularly important in early detection where potential biomarkers are likely to be low abundance. Since it's non-invasive, high-frequency serial sampling is possible with no ethical or patient comfort concerns. As I mentioned earlier, breath is for more than just respiratory disease. Breath could also be used as an orthogonal approach to other biomarker platforms. And lastly, because breath is so easily accessible, there is the potential to translate to point of care or even home-based testing in the future. Where are we currently in breathomics? Modern breath testing started with Linus Pauling back in 1971, and over the last 50 years, there have been hundreds of scientific papers suggesting the presence of VOC biomarkers across a range of diseases. One thing that becomes clear in going through these papers is that there is little agreement in biomarkers associated with particular diseases. That probably answers the puzzling question of why breath testing is not yet used routinely in clinical settings. Another thing that becomes clear is that there was no standardization of breath collection and analysis. These homebrew methods really lack the hardware and protocols necessary for robust, repeatable breath sampling that is necessary for confident biomarker discovery and validation. This challenge is what we have addressed with our breath biopsy system, which allows for the first time the reproducible collection and analysis of breath samples. The collection side consists of the Receiva breath sampler, the Casper portable air supply, and the breath biopsy collect software. The analysis side consists of a high resolution, accurate mass, gas chromatography, mass spectrometry system. I will discuss each of these components in a bit more detail over the next few slides. Receiva is the first standardized device that enables patient breath samples to be collected reliably and reproducibly. During sampling, patients just breathe normally while Receiva tracks their breathing pattern. This is done with pressure and CO2 sensors that you can see in the schematic. At the appropriate breath fraction, pumps are activated and pull the exhaled breath onto the sorbent tubes. Over a period of time, Receiva traps and stabilizes the VOCs and pre-concentrates them onto these tubes, which are a bit like a vacutainer for breath. These are then sent to our breath biopsy lab for analysis. Receiva has been deployed in studies at over 100 clinical sites and hospitals around the world, including the world's largest breath-based clinical trial. Here's what a breath collection looks like. The Receiva has CO2 and pressure sensors that monitor tidal breathing in real time, shown here at the top. The Receiva also has two pumps, so what you see in the middle is that during expiration, the pumps are being activated sequentially. So in a single breath collection, there are two different breath fractions captured, alveolar and bronchial. The pumps are programmable, so it is possible to collect the whole breath sample. Receiva collects a set volume of breath, which takes approximately 10 minutes. Since our blood volume circulates through our lungs about once per minute, there's quite a large headspace of blood collected in a given sample, and this pre-concentration enables much higher sensitivity. Another component on the collection side is the Casper portable air supply. A real challenge in breath analysis is the inclusion of environmental VOCs in breath samples. These can come from things like air pollution or cleaning products and can be at much higher levels than endogenous VOCs, so they can interfere if included in a breath sample. Environmental VOCs also vary over time and across clinical sites, making inter-site comparisons difficult. And consider that the room where the breath sample is collected will contain VOCs from any other people breathing in the room. It won't be possible to know if a VOC is a true on-breath compound or something coming from the environment. Until we developed our breath biopsy system, breath samples were typically captured in bags. Patients would inhale air from the room and then blow into the bag. These samples contain a lot that isn't really representative of a patient's phenotype, and this is probably why many previous breath studies have not been reproduced in terms of the biomarkers identified. Casper filters the air patients are breathing, which significantly reduces contamination by environmental VOCs. The scrubbed air is supplied by a tube that goes from Casper to Receiva. The graph on the bottom left shows a comparison between a sample collected with and without Casper. With Casper, there is a significant reduction in the overall signal coming from the environment. In the middle graph, you can see that with a clean background from Casper, the sensitivity is much higher than with a contaminated background, where there is little difference between the VOC level between what is on breath or from the environment. On the right, you can see how Casper reduces false discovery, as this VOC is clearly not on breath. It is coming from the environment. So the combination of Receiva breath sampler and Casper portable air supply allows for the reproducible and reliable collection of breath samples. The final component of our breath biopsy system is a standardized analysis platform we have developed, which includes the gold standard for biomarker discovery, thermal desorption, gas chromatography, mass spectrometry. We use a high resolution, accurate mass spectrometer that has been optimized to enable high quality VOC detection. The high resolution maximizes the potential for detecting a wide range of VOCs covering many chemical classes, and the sensitivity allows for detection of low abundance biomarkers. Although HRAM, or high resolution accurate mass gas chromatography mass spectrometry, is a gold standard for biomarker detection, it does require standardized protocols to ensure reproducibility and confidence in the data generated. We've developed our Omni assay with this in mind. A key feature of this is our breath biopsy HRAM library, which consists of VOC standards that we have run across our platform. Since we know the signature of each VOC standard, we can match it to the VOC signal in a breath sample and make a high confidence call on each VOC identity. For many of these VOCs, we may know the biological origin or function, which allows us to build targeted panels in specific disease areas. Here's an example of a targeted panel. This is our respiratory panel, and it contains VOCs previously implicated in chronic inflammatory diseases of the lung. So what does a breath biopsy study look like? We will provide a collection station at each clinical site, along with online training and support throughout the trial. We will also provide a regular supply of breath biopsy kits. Each kit contains a single-use mask and the breath biopsy cartridge for collecting a breath sample. Samples and subject data get collected in your clinic. The subject metadata then gets securely transferred to our breath biopsy digital biobank, and the cartridges get shipped to our lab at ambient temperature for analysis. Shipping on dry ice is not required. We will then analyze the samples, and depending on the project needs, we can provide comprehensive statistical analysis and biological interpretation. Final slide from me. We will be holding our 4th annual breath biopsy conference this year on October 12th and 13th. The purpose of this conference is to facilitate progress in the breath research field by bringing together leading researchers and research organizations to share the latest advances and discuss common challenges. The key areas of expertise in and around the breath research field will be explored in talks, poster sessions, and discussions. The conference will be virtual and is free to attend, so we look forward to your participation. Registration and abstract submissions are open now. And now I'll turn it over to Rob Money, who is Owlstone's Head of Clinical and Translational Science.